This is episode 2. This is a series where I, vi.amara, the Sustainable Lab guest promoter, interviews people who are interested in integrating sustainability into science. Today I'm joined by Eric Kalkman. Thank you so much for joining me. Would you like to start by introducing yourself?
Yes, thank you. Yes, I'm lab manager in the Helgason Leukemia Research Group at the School of Cancer Sciences, and we recently resubmitted for the LEAF Silver Award again. We're quite a translational lab. I work on improving patient drug treatment and I work on a variety of leukemias.
That's great. So, why is sustainability important in general? Why do you think so?
So when you sit back and objectively look at how much single-use plasticware the average research lab uses, it's pretty staggering. And so, as I found as well, is cutting back on single-use plastics, reuse, and generally aiming to be more environmentally friendly, really can go hand in hand with saving money as well in the research lab. And it just requires the willingness and cooperation of the team from the top down.
Yes, that's right. Keeping in mind your specific lab group, is there anything specific that pushed you to sign up for LEAF?
So I think researchers in life sciences are possibly more acutely aware that we're really part of a wider society, and as such really have the responsibility not just for our research area but also for doing our bit for the planet. And so I've always found that when you do your bit at home with recycling, if you, like I said, if you look at labs, they're very environmentally unfriendly historically, moving away from a lot of glass to single-use plastic. And so just doing what you can hopefully make some impact, definitely university-wide.
What LEAF awards have you been awarded?
So we went for bronze to start with, and we've gone, so this is the second or third time we've applied for silver now.
Do you have any plans on applying for the gold anytime soon?
I will have a look into it, but I think going for bronze is definitely easy to accommodate in the research lab. Silver is still doable, but gold might be a bit more challenging because there's only so many things you can really change in your usage.
Yeah, it's hard to find a balance of being able to satisfy all of the criteria and being able to do your actual research.
Yes, yeah.
But yes, was there anything specific that was innovative, or that's something that you're proud of that you did to satisfy the LEAF criteria?
So not necessarily to satisfy the LEAF criteria, but not too long ago I introduced what's called Aquatech water preservation cell in our water baths in tissue culture. Historically, you would use biocidal liquids in the water baths that we use for heating up growth medium for cell culture, but these are now no longer allowed legally. And we were finding that we had a lot of recurring infections in our tissue culture rooms, and we couldn't solve this until I introduced these water cells which release silver into the water, silver ions, and therefore are also biocidal but more environmentally friendly. And this has really reduced the number of infections, so it's definitely working. But it did involve me convincing 6 individual PIs to contribute towards the cost of this. And so it was a challenge, but it paid off, which I'm quite happy about.
Sounds really great, yeah, good job. To contrast that question, was there any criteria that you find particularly hard?
The one criteria that I find hard is cutting electricity usage, like turning off PCs, screens, equipment that isn't in use, but also moving from ULT freezers, older models, to newer, more eco-friendly models. It's there's not a direct financial benefit to PIs to promote this or to push for this. And I think that probably university-wide, this is the biggest thing that they could save on, both financially as well as for the environment. But yeah, it's hard it's a hard sell because there's no direct benefit that you can show for this. So I try my best in our lab, but I still struggle to get people to cooperate in that respect. There's also it would be great if there, for example, was funding available within the university to move away from very old freezers that are much more heavy on electricity use towards newer models. Like these ULT freezers are like £10,000, for example, so that not every research lab can like move on their own. There's not always budget for that.
Yeah, that's definitely something that uni can help with.
Yeah.
Are there any non-environmental benefits of leaf that you've encountered?
So like I said, I've always been trying to get sustainability as a byproduct of cost saving, so to make it more attractive to the PI, to the lab head, and so there's a lot of things that you can that are both help sustainability but also like financially more interesting. Like what we've done in our lab is move from plastic, like disposable scalpels, for example, back to the more old-fashioned, like when you buy a handle and you replace just the blade, which is obviously no plastic involved in that, so it's much more better for the environment, but it's also financially better because they're cheaper in the long run. Also, another thing that we've done, for example, is that we use a lot of tubes for flow cytometry, a particular assay, and we used to get these tubes directly from stores, from university stores, with lids that we then immediately discarded because we didn't need those. And now I order these externally without the lid, and not necessarily that they're cheaper, but at least there's a lot less plastic waste.
That's good. Do you have any tips for students to be more sustainable in the labs?
Yeah, so it really pay attention to detail. There's any number of ways you can reduce plasticware and like make your lab more environment friendly. Like I was saying, like for example, tips. You can if you do a zero dilution, you can reduce tips on that. Instead of using like a tip for every row, you can use a single set of tips. But also looking at required waste streams, for example. There's a lot of waste put into bins that are then for special disposal, that they need to be incinerated, for example. Not every waste needs to. There's a lot of wrapping material in plastics that you can actually recycle. A lot of, for example, media bottles that you can rinse and then recycle instead of just binning them and having them go to waste.
Yeah, small things like that do add up.
Yes,
like,
yeah.
I have 40 people in my lab itself, and trying to even think about to scale that up. There's like 600 people in the whole biology course right now. So the amount of waste we produce in a single lab session is not the best.
Yeah, so it's really about reassessing your plastic use and how you do things in the lab, and really be proactive, but there's always going to be a compromise as to how much you can achieve, and so you have to be realistic as well. Not everything's feasible. Like moving, for example, back to glass serological pads is not possible because there's not enough washing facilities, definitely not in this building, for example. So you can't move away from plastic there, but just be sensible and try and reuse what you can.
Exactly, yeah. Just to finish off, is there anything you would like to say to the labs that have not yet signed up to leave?
Yeah, so I think it's not as hard a task as it might seem when you start. Like when you go for bronze, it is a bit of work, but it's rewarding to know that you're at least doing your bit for the planet, and also, as I said, there's ways that you can marry it to saving costs as well.
Yeah, exactly. So thank you so much for joining me today, and thank you for watching.